The overall goal of this method is to rapidly synthesize and purify Trehalose analogues, which have possible applications ranging from the detection of pathogenic bacteria to the preservation of biomaterials.
This is a fast and high-yielding method that provides access to analogues of the disaccharide Trehalose. These compounds have various established and emerging applications in biomedical and biotechnological research. This method consists of a one-step enzymatic synthesis and an all-aqueous purification. Together, these procedures help to address some of the drawbacks of traditional chemical syntheses of Trehalose analogues, which can be lengthy, low-yielding, and environmentally unfavorable.
The implications of this method may extend toward the study and diagnosis of tuberculosis, because modified Trehalose analogues have been used for imaging Mycobacterium tuberculosis, the causative agent of tuberculosis. This method also can help accelerate the synthesis and evaluation of Trehalose analogues for other possible applications, such as preservation of biological products and use as non-caloric sweeteners.
Prior to beginning the procedure, prepare an overnight culture of Tre-T expressing E. coli. To begin the Tre-T expression procedure, place 750 milliliters of TB growth medium in a 2,800 milliliter Fernbach culture flask and autoclave it. Add ampicillin to the broth to achieve a final concentration of 100 micrograms per milliliter. Obtain cuvettes for UV vis spectroscopy and fill one cuvette with broth from the flask as a blank. Next, add 3 milliliters of the prepared Tre-T expressing E. coli overnight culture to the flask. Incubate the flask at 37 degrees Celsius while shaking at 200.